We're going to talk about the central nervous system, and we'll talk about the basic determinants of CNS physiology. We're going to first start off talking about the glucose requirements and the oxygen requirements of the brain. We can see that the brain requires approximately 80 milligrams of glucose per minute and approximately 50 mLs of oxygen per minute. So assuming that the brain requires and receives about 80 milligrams of glucose and 50 mLs of oxygen, we can say that through a process of glycolysis and TCA, the brain will extrapolate about 38 ATP for every glucose molecule that it oxidizes. And we can think of ATP as an energy source within the cellular activity. And how is that energy source used or utilized within the CNS system? We can say about 60% of the ATP that's being produced through glycolysis and TCA can go towards the neuroelectrical activity within the brain tissue. And about 40% of the ATP that's being utilized goes towards the cellular integrity within the brain tissue. And to show and illustrate how this is being used, we can look at the single neuron to depict how and what takes place in the neurophysiology. So first thing to notice is the concentration gradient. We can say that the concentration gradient outside of the cell or extracellularly, there's a higher sodium concentration outside or extracellularly compared to the intracellular sodium concentration. And because of that, there's a higher positive charge outside or extracellularly. And you can see that as the relatively positive charge at the outer cell membrane structure here. And relatively inside, it's a relatively negative charge in the inner aspect of the cell, right next to the cell membrane. And if the brain were to be stimulated, if the dendrites were stimulated or some receptor activity were to be stimulated on the cell, and it activates the sodium channel, it's a voltage-gated sodium ion channel, it allows sodium to enter the cell. And as sodium enters the cell, the charge depolarizes the cell membrane, in which now there's a relatively positive charge in the inner aspect of the cell, or intracellularly, and a relatively negative charge outside the cell. And that basically propagates an action potential down the axon, and it travels down the axon until it gets to the nerve terminal. And at the nerve terminal, there's a calcium-regulated response in which neurotransmitters within a vesicle are released into the synaptic cleft. The synaptic cleft is basically the space between the nerve terminal and the next cell that will receive the signal via receptor and the neurotransmitter. So the neurotransmitter that's released from the vesicle of the nerve terminal is released into the synaptic cleft, and the nerve or the neurotransmitter will go to the respective neurotransmitter. For example, if norepinephrine were to be released, norepinephrine will go to its respective receptor and create a conformational change in the cell membrane, and a certain physiological response will take place within the cell. That's the normal neurophysiological response within a cell once it's stimulated. And we can say basically as the sodium goes down its concentration gradient, meaning a high concentration gradient of sodium outside the cell, down its concentration gradient once the voltage-gated ion is opened, the channel, it allows sodium to easily come into the cell. And we're going to later talk about how once the repolarization takes place within the cell membrane, meaning that the charge becomes reestablished in which the positive charge is outside the cell membrane and there's a relatively negative charge within the inner aspect of the cell membrane. That's considered repolarization. But the component that we're going to look at is how does the sodium concentration that starts to increase intracellularly as sodium enters into the cell membrane, or rather passes through the cell membrane during depolarization, how does that process take place in which the sodium is pumped back outside the cell against its concentration gradient through a process of sodium-potassium ion pumps? And we'll see that basically it requires energy, and that energy is in the form of ATP. So if we were to look back at the beginning premise, and we say that the brain requires approximately 80 milligrams of glucose per minute and requires about 50 mLs of oxygen per minute, that will allow the brain tissue to use the process of glycolysis and TCA to form ATP, approximately 38 ATP for every glucose molecule that's being oxidized. And that energy is used for the neuroelectrical activity and maintaining cellular integrity. And that neuroelectrical activity we can focus on the sodium-potassium pumps. So out of that 38 ATP, it's going to be used towards the ATP that's required in order to pump the sodium against its concentration gradient, meaning there's a higher concentration of sodium outside the cell, and in order to push or pump that sodium out, it requires energy, and that energy is ATP. Okay? So the whole focus that we're going to focus on, or the premise that we're going to focus on, is what if we don't have enough ATP? What happens in this process here that leads to the demise of the brain tissue? For example, the increased sodium concentration that starts to build up intracellularly, and unable to pump it out because we don't have the ATP, sodium starts to build up, and eventually water is going to be able to pass through this membrane because of the increased sodium concentration. There's going to be an oncotic pressure that draws the water in, and there's going to be a cellular swelling that takes place within the nerve tissue called cerebral edema. Likewise, when you don't have the energy to maintain the cellular integrity of the brain tissue or the cellular membrane, you're going to start to get leaky cell membranes that will even allow more sodium to enter the cell. And again, that's going to create worsening cerebral edema. So obviously this takes place when the brain does not receive adequate amount of oxygen and glucose to the brain. And the way that the brain receives these major components is through the blood. So we'll focus on what factors regulate the cerebral blood flow in order to supply the brain with adequate amount of glucose and oxygen so that all this cellular activity can take place to create the energy, which is ATP, to maintain the neuroelectrical activity and the cellular integrity of the brain tissue. So in the next lecture, we'll focus on the major factors that govern cerebral blood flow. And the major influencing factors of cerebral blood flow, which we'll talk about, is the cerebral perfusion pressure, autoregulation, and certain extrinsic factors such as gas tensions, temperature, viscosity, and autonomic influences of the body that regulate cerebral blood flow.